In this video, we will study about transcription in eukaryotes. I've already posted a video on transcription in prokaryotes. The link is in the description box. If you haven't watched it, then you watch that video first so that you understand this video better. But still, we will go through the basic points because there is not much difference between the transcription of prokaryotes and eukaryotes with little differences. So let's first go through the basic steps. What is transcription? The definition is that the process of copying genetic information from DNA to RNA. So if RNA is formed from DNA, that is called transcription. The location in eukaryotes, it occurs in the nucleus because eukaryotes have a well-defined nucleus and DNA is present inside that, and so DNA forms RNA inside the nucleus. Whereas in prokaryotes, it occurs in cytoplasm. Then what is central dogma? Central dogma was given by Francis Crick, and he said DNA first forms RNA by transcription, and then from RNA proteins are formed by translation. So this is a one flow direction in a human body, that is DNA will form RNA and RNA will form protein. So this is central dogma which was given by Francis Crick. Then he said that there are two conditions for transcription. Transcription occurs first of all only in a segment of DNA. It does not occur in the entire strand of DNA like how replication occurs in the entire strand, but transcription occurs only in a segment of DNA. Then second point is that only one of the strands of DNA is copied. Both the strands are not copied. What is the reason behind it, that why one of the strands of DNA is copied? Because if both the strands of DNA will be copied, RNA which will be formed will be complementary to each other. So if we look at this DNA strand and we can see the bases, so if RNA is formed here, instead of we know that there is no thymine in RNA, instead of thymine there is uracil. So here there will be uracil, here adenine, guanine, cytosine. So if we consider just these four bases, and on the other strand if we consider it is A, this will be uracil, C and G. So we find that the RNA, if it is formed from both the strands, then they will be complementary to each other and they will bind together. So if they bind together, it means this RNA which will be formed will be a double-stranded RNA, and so from double-stranded RNA protein cannot be synthesized. So central dogma is not going to be followed. So this is one reason that why both the strands are not used. And the other thing is that naturally if both the strands are suppose forming different RNA, means they will code for different proteins. So from one strand of DNA, or not one strand, one molecule of DNA, if there are two different proteins formed, then that is not acceptable. So that is why only single stranded RNA has to be formed, and that is why only one of the strands of DNA is copied. The first point was that the transcription occurs only in a segment of DNA. So when we look at this figure, this is a DNA molecule, and only a segment of DNA which forms RNA, that is called structural gene. So it is only this portion, this much portion, which will actually form RNA. The complementary bases will be added. It starts at a promoter region and ends at terminator region. So what happens is that RNA polymerase, the enzyme, it binds to the promoter site, copies the complementary strands here at the structural gene, and then by the time it reaches the terminator, then it drops off and RNA is released. So only a part of DNA forms RNA, that is the structural gene only forms RNA, and only one of the strands of DNA is copied and it forms RNA. Now the enzyme that is used for transcription is RNA polymerase, and it makes RNA strand from 5' to 3' direction. So it means the DNA strand on which it will make this strand should be in 3' to 5' direction. It means this strand, on this strand RNA will be formed. The strand which is from 3' to 5' direction is known as template strand, and the other strand is known as coding strand. Now what happens, we are still talking about prokaryotes, that when RNA polymerase comes and attaches itself to the promoter site along with sigma, then it starts adding the nucleotides and mRNA is formed from 5' to 3' direction. And when it reaches the terminator site, it attaches to the Rho factor, and then this RNA strand is released. This is how it is formed. When we look at the bases of RNA, we find that because this is G, it's going to be C, G, A, and instead of thymine, there will be uracil here. We find that the mRNA sequences are complementary to the template strand, whereas they are exact copy to the coding strand. Only the difference is of thymine and uracil, because it is RNA. Instead of thymine, there is uracil, otherwise the sequence is same. So if it is asked, it's often asked in the paper, that the mRNA which is formed, is it complementary to template strand or coding strand? So it is complementary to the template strand, and it is exact copy of the coding strand. Now let's talk about the differences between prokaryotes and eukaryotes, and then we'll understand the process of transcription in eukaryotes. Okay, now let's understand the differences in transcription between prokaryotes and eukaryotes. We already discussed that in prokaryotes, the transcription occurs in the cytoplasm, whereas in eukaryotes, it occurs in the nucleus. In prokaryotes, there is just one enzyme, that is RNA polymerase, which is used for transcription, whereas in eukaryotes, there are 3 RNA polymerase. RNA polymerase I forms ribosomal RNA. There are 3 types of ribosomal RNA, 28S, 18S, and 5.8S. RNA polymerase II forms messenger RNA, whereas RNA polymerase III forms tRNA, then 5SR RNA. SR stands for small regulatory RNA, and snRNA, which is known as small nuclear RNA. So there are 3 types of RNA polymerase for eukaryotes: RNA polymerase I, II, and III. 1 makes ribosomal RNA, 2 makes messenger RNA, and 3 makes tRNA. Then 5SR RNA and snRNA. So you should know all these different types of RNAs that RNA polymerase makes. You can also remember that RNA polymerase I makes ribosomal, second makes messenger, and third makes transfer RNA. So RMT, first makes R, second makes M, and third makes T. So you can remember it like that. Then the next thing is that for prokaryotes, for transcription, sigma factor and rho factor are required. Sigma factor is required for the initiation of transcription, and rho factor is required for the termination of transcription. Whereas in eukaryotes, these factors are not required. They can be the messenger RNA or any kind of RNA can be formed in without sigma factor or even without rho factor. So these are the 3 differences between prokaryotes and eukaryotes. Now let's understand transcription in eukaryotes. We know that there are 3 types of RNA polymerase in eukaryotes. RNA polymerase I makes ribosomal RNA, RNA polymerase II makes messenger RNA, and RNA polymerase III makes tRNA. So let's first discuss that we are making a ribosomal RNA and tRNA. So what happens is that RNA polymerase, whether it is 1 or 3, it moves along the DNA strand, and when it reaches the promoter site, it comes to know that it has to make RNA. So it starts making RNA in 5' to 3' direction where the structural gene is present. We know that it will be complementary to the template strand. So the bases are here G, so it will be C, this will be G. Instead of thymine, there will be uracil, A, this is uracil, and this is G. So this is this is RNA which is formed. We are talking about ribosomal RNA and tRNA. As soon as this RNA polymerase will reach the terminator end, then what will happen? It will release the RNA strand. So what happens is when it attaches to the promoter site, then both the strands of DNA separate and they start forming RNA. When it reaches the terminator site, RNA polymerase is released and both the strands of the template strand of DNA and RNA, which were binding together because RNA polymerase is released, they are also free and then RNA is released. Now this process occurs inside the nucleus in eukaryotes, because eukaryotes have nucleus, and from DNA this RNA is produced and it occurs in the nucleus. In case of ribosomal RNA and tRNA, as soon as this RNA is formed in the nucleus, after that it leaves the nucleus and it goes into the cytoplasm where it has to do translation. So that happens in the case of RNA and tRNA. But in case of mRNA, which is made by RNA polymerase II, so if it is RNA polymerase II, then mRNA is going to be formed. But this mRNA is not known as mRNA right now when it is formed. It is known as HNRNA, which is formed by RNA polymerase II. HN stands for heterogeneous nuclear RNA, or it is also known as pre-RNA and also called precursor RNA. So this RNA, which is formed by RNA polymerase II, is known as hnRNA. It is not called mRNA right now because it has to be processed, and all this is happening inside the nucleus. So this processing, that is when it has to convert hnRNA into mRNA, will also occur inside the nucleus. After it is converted into mRNA, then it will go into the cytoplasm. So this is the main difference, that when RNA polymerase I or 3, when they are making ribosomal RNA or tRNA, as soon as they are formed, they're already ready to do translation. So they leave the nucleus and go into the cytoplasm. But when RNA polymerase forms RNA, this messenger RNA is not processed because it is not messenger RNA right now. It is hnRNA, heterogeneous nuclear RNA, or pre-RNA or precursor RNA, which is not ready for translation. So that is why it has to be processed, and then it converts into messenger RNA. After that, it will leave the nucleus and go into the cytoplasm for translation. So now let's understand what is hnRNA, what is the structure, and how it is converted into messenger RNA. One more point to be mentioned, that I said that as soon as RNA polymerase I, 2 or 3, whichever binds to the promoter site, then the DNA strand is separate. It's not the entire DNA strand, it is only that portion where RNA polymerase is attaching to the DNA strand on the template strand. There the strands separate and form a kind of bubble, and RNA is formed, whether it is rRNA or tRNA or mRNA or hnRNA. So whichever RNA is formed, only a bubble is formed in the double-stranded DNA molecule, so that the two strands separate at that particular point and RNA is formed. After that, as soon as it reaches the terminator, then RNA polymerase drops off, and when it drops off, the RNA which was binding to the template strand, because of the complementarity, they separate, template strand and the RNA strand, it separates, and then RNA is released from that strand. So it is not the entire strand which separates in DNA, but it is only that particular point or particular area where RNA polymerase is attaching and moving. Now let's understand about hnRNA. When RNA polymerase II attaches to the DNA part, to the promoter site, and then starts making RNA when it enters the structural gene, then the RNA strand is formed. We know that the RNA strand is formed from 5' to 3' direction. It means this 5' end is going to be formed first. When it reaches to around 20 to 30 nucleotides, then a process called capping occurs at the 5' end. So here capping occurs. This is called capping. Now what is capping? It is addition of guanine nucleotides. These guanine nucleotides are added. These are also known as methyl guanosine triphosphate. So these guanine nucleotides, or we can say methyl guanosine triphosphate, are added to 5' end of the RNA. This RNA is called hnRNA right now, heterogeneous nuclear RNA or pre-RNA or precursor RNA. Now, as soon as the 3' end is reached to whatever level it has to reach, and then this RNA strand is released into the nucleus. Now, this 3' end, when it is released, there is another process which occurs, which is called tailing. So, here already capping is done, here capping is done, and as soon as this RNA is released, then tailing is done here. Now, what is tailing? Tailing is addition of adenine nucleotides. So, here adenine nucleotides are added. Adenine nucleotides. This process is also known as polyadenylation. Polyadenylation, because many adenine nucleotides are added, and those nucleotides are around 200 to 300. You have to remember this number. So, at 3' end, tailing is done as soon as the RNA is released into the nucleus, and 5' end capping is done before, much before, because around 20 to 30 nucleotides are formed. Then capping is done, which is done by guanine nucleotide or methyl guanosine triphosphate. We can also write methyl guanosine triphosphate, like this we can write, and here we can write A, polyadenylation. So, this is poly A tail also, it is also called poly A tail. Now, what is the purpose of capping? Let us first understand that. Capping is done so that this end of RNA is not hydrolyzed by the exonuclease enzyme present in the nucleus. One reason is that, and the second reason is that the ribosomes, when this RNA is going to enter the cytoplasm and reach the ribosome, then ribosome identifies this RNA and attaches itself from the 5' end because of the presence of these guanine nucleotides, because of this cap. So, this is the reason of capping, and tailing is done mainly for the same reason, to protect the tail or protect the end of the RNA from hydrolysis by exonuclease. Now, why is it's not done in tRNA or ribosomal RNA? Because when tRNA or ribosomal RNA, when they are formed in the nucleus, they are already ready to leave the nucleus and enter cytoplasm and start doing translation. But that is why they do not need any protection. But this hnRNA is not ready, mRNA is not yet formed. That is why these ends have to be protected by the time it reaches the cytoplasm. So, one thing is this capping and tailing. Second thing is that this hnRNA has coding and non-coding region. Coding region is the one which will code for protein during translation, and non-coding region is the one which does not code for any protein. So, on hnRNA, the non-coding region which does not code for any protein is called introns. So, here I have marked it in blue. These are introns. This is also intron. These are all introns. They will not code for any protein. So, because they do not have they do not code for any protein, they should be removed. The portion which codes for protein are called exons. So, this red portion is exons. So, this is also exon, then this is also exon. Wherever the red strand is, that codes for protein. So, these are exons. So, the next process is done, which is known as splicing. Now, what is splicing? Removal of introns, because we do not need them. So, removal of introns and joining of exons. This process is called splicing, and it is done by a structure which is called spliceosome. spliceosome. Now, what is spliceosome? They are SNPs. SNPs means they are small nuclear ribonucleoproteins. So, these structures are called spliceosomes, and they go and attach on wherever the introns are. They will remove these introns and then join the exons. So, finally, hnRNA is processed and we have now messenger RNA. What does it have? This is messenger RNA, which has 5' end and 3' end. At 5' end, it has methyl guanosine triphosphate, the cap, and then it has a poly A tail. Only exons are present in it, introns are all removed. So, this is finally a processed hnRNA, and this is full. Now, this is a messenger RNA because finally it is processed. We will not call it hnRNA. This messenger RNA is now ready to leave the nucleus, go into the cytoplasm and start translation. One more thing is that when we say that what is the, is there any difference in the size of hnRNA and messenger RNA? So, we will say yeah, messenger RNA is smaller than hnRNA because from hnRNA introns are removed, and this messenger RNA does not have any introns. That is why this messenger RNA is smaller than hnRNA. So, this completes with the transcription in eukaryotes. It's quite similar to that of prokaryotes, only the difference is that they have these 3 types of enzyme: RNA polymerase I, 2 and 3, and there is a difference in how messenger RNA is formed. So, I hope you like my videos. If you like my videos, then do subscribe to my channel, share and leave a comment of any portion you found more difficult and which other topic should I teach. Thanks for watching.